Well, thank you for inviting me. I cut a few slides so that I could talk a little more about what was here and what we heard today. And I'm really honored to introduce you to the Neuro and Immune Institute in Reno, Nevada. What you see here is an architect's drawing of the Institute upon completion in 2010. And you know that my first introduction to Nevada was that this was from Las Vegas, because nowhere do we put Porsche in our facilities at the National Institutes of Health in the artist's rendition. It wouldn't go well with the government. So what is our mission there at the Institute? Well, our focus is neuroimmune diseases. We've heard a lot today about ME/CSF. We've also heard about all other types of neuroimmune diseases, atypical MS, fibromyalgia, autism, which we haven't said a lot about, but there's interesting new data emerging from the Mind Institute in Sacramento, California, and others. And what we found is that these diseases involve, and we've shown here today, complex immune abnormalities affecting the function of both the body and the brain, and that these can result in lifelong disability. So our focus is primarily to advance patient care, to train medical students and physicians. So Andrew, we have a job for you in a few years when you finish medical school, because you'll be perfect for understanding our group. We're researching the pathophysiology of these diseases, and finally, to be fully translational, to develop therapeutics, diagnostics, and prevention strategies. My other hat is in drug development as well as basic cancer research. So when I came to the Institute 2 years ago, I initially met Dan Peterson, and he had been studying a cohort in Nevada for more than 25 years. Since 1983, this outbreak was identified, and Dan identified the clinical priorities for the Institute based on these criteria. So we wanted to establish genetic signatures of disease subsets. And when a year or so ago, I met Jonathan Kerr, and he showed us his expression data, and we immediately began to collaborate with him in that endeavor, because we don't want to repeat that. Now, we are looking at the genome level for polymorphisms and differences, and such that you might be born with a genetic dysfunction in a gene such that it's has to be changed by gene therapy, or you have to know that it exists so that you understand the basis. We're also looking at microbial co-infections. Our cohort has a lot of herpes virus 6A, which is distinct from 6B but in its neurotropism and in biology, but I won't go into it, and novel viruses, which I'll discuss later. Our goal is to identify and validate biomarkers, serum biomarkers, urine biomarkers, CSF, things that we can do to immunologically profile the patient as well as virologically profile the patient and develop therapeutic algorithms. The Whittemores have purchased a diagnostic company which is currently operating as a company called Viral Immune Pathologies DX, and they intend to integrate that into the Institute as a wholly owned subsidiary of this nonprofit, such that IP, intellectual property, and things that are created within the Institute, within our collaborations, can be translated back to the patient so that we can treat more patients. So importantly, a major part, and you heard John Chia say quite nicely today, that patient samples, in order to do these investigations, are really everything. Control of those patient samples, standardizing how they're drawn, where they're drawn, how long before we make RNA, and how we stabilize the various nucleic acids in sera are very important. Dr. Peterson was prescient in the early 80s in purchasing a -80 freezer and collecting blood samples, making peripheral blood mononuclear cell pellets of every patient every time they came in, cerebral spinal fluid, you name it, it's there. It was in this -80 freezer when I got there 2 years ago in Ziploc bags and tubes. So we have since put to work 3 medical students to create a fully categorized and searchable database which shows the clinical data. They read through the charts and correlated it, entered the data into a database, entered every test that came back from a diagnostic company, every test that came from the research, and where each sample was, and now it is currently a fully searchable database that we can share with investigators that maybe have these samples that have criteria that we can look at, and we can collaborate to get to answers faster. We purchased also a 25,000 capacity liquid nitrogen cryostorage freezer, as well as 2 additional -70s and a -150 freezer. Along with that, we have a fully functioning bioprocessing lab that we operate as a biosafety level 3, because we've heard a lot about the pathogens today, so we're extremely cautious about the safety. And that lab is up and running right now, as is our research program. So our current research strategies are really working on the central hypothesis, which is one that our collaborator Paul Levine, who we heard about earlier today, and John, you showed a much nicer slide than I have on this hypothesis, but the idea is that chronic inflammation and stimulation from both acute infections and what I call reactivated systemic infections. We all harbor EBV, a lot of these CMV enteroviruses, and we're not sick. So why? What tilts the balance of that? We call it homeostasis. What disrupts the delicate balance of the immune system? That's what we're investigating. And what's a susceptible host genetic background, environmental background? What's the trigger that leads to that pathogenesis that we characterize as MECSF? And we think we'll find answers to those other neuroimmune diseases we talked about in focusing here on this cohort. So Jonathan, I mentioned, has done the gene expression profiling, and he will look at his subgroups with our patient samples such that we can identify which subgroup, if there's any single subgroup, and I'm guessing there is, that our cohort, which is very tightly controlled and similar, will fall into. I'm going to show you novel virus identification in MECSF through really state-of-the-art microarray technology. I'm going to show you how we cytokine and chemokine profile in a multiplex mixture where we look at more than 25 immune factors, cytokines and chemokines, simultaneously in a single 50 microliter sample of blood. I won't show you today the immunological profiling done by flow cytometry where we look at the cells, but the Whittemores purchased 2 5-color flow cytometers as well as a 15-color state-of-the-art flow cytometer and a Luminex instrument for complete immunological profiling. I should say when they started this endeavor that they engaged the state and federal governments prior to even hiring me, and they have gotten through persistent lobbying support, annual support from the state government and instrumentation support in $1,500,000 this year from the federal government for the purchase of these instruments. We will talk briefly about innate immune defects, and I did slip these slides in when John mentioned RNA cell, because that's a major defect and we might have some answers there for him. And we're going to look specifically at something called a clonal T-cell receptor gamma rearrangement and the mantle cell lymphoma development in a subgroup of Dan's cohort there at the Institute. So we've heard a lot today, so I won't belabor this slide, about the background of the idea that infectious agents can indeed trigger and perpetuate MECSF. Not only viruses, the herpes viruses, but we've heard about Borrelia, the enterovirus, mycoplasma, and parvoviruses. All are reasonable players. Lenny asked me who won. I'll tell you that in a minute. Actually, nobody has. It's dependent on the patient. So again, the hypothesis is co-infection of microbial pathogens, unknown viruses, 6A because that is the key virus in our cohort, but by no means is that the only trigger of the inflammatory response, resulting in a chronically dysregulated immune response. And we are confident that by profiling the cytokines and chemokines and identifying these abnormal immune subsets of cells early, that we can reset the rheostat or the thermostat such that we can get the immune system with anti-inflammatories. Jean talked about immunomodulatory drugs. We can actually get the desired immune response and bring back the function. So with that in mind, we talked a lot today about innate immunity. So this slide really depicts, this is the last 5 years. So we didn't know anything about what we call these pattern recognition receptors here, the toll-like receptors that were identified in Drosophila. These recognize, this is the innate immune response. Malcolm talked early this morning, and as did Irving, about the immune system. So there's a war going on. This is the front line. They see viruses through toll receptor 3, these endosomally maintained receptors. We've heard about toll receptor 4, which see pathogens, LPS, endotoxin, gram-positive and negative. They see everything, and they're very specific. But what's interesting is they take these signals at the cell surface or in the endosome for viruses that might be endosomally maintained. They don't have to get outside the cell and come back down and signal. We can see those RNA that John described for enteroviruses. We see that quite best by the double-stranded endonuclease known as RNA cell, and I'll talk about that later. It's not on this slide. But importantly, all of those signals are integrated to lead to the production of cytokines. So NF-kappa B was the major thing identified in Jonathan's overexpression gene. That's a response to pathogens. It quickly upregulates pro-inflammatory cytokines and chemokines. And when it's not held in tight check, it can cause inflammatory disease. There are other cytokine repertoires and chemokine repertoires that we can identify. We can look at interferon beta and rantes and consider who the pathogen might be by the profile that we see on expression. And certainly to modulate interferon alpha and beta in maintaining the antiviral properties of these important cytokines. So the first thing we asked was, what's the role of novel viruses and co-infections in ME/CFS? And to do this, we used a custom mammalian virus microarray, and we looked at our cohort of patients with these clonal rearrangements and these lymphoid malignancies. Now, I'm going to describe this microarray in a little bit more detail because it was originally used maybe a year before I came to the program, because remember, I worked in cancer. And prostate cancer and breast cancer are known to be inflammatory endocrine-driven tumors. The hereditary prostate cancer gene was identified upon sequencing the genome as RNase L. So what the group in Stanford did was they constructed a 10,000 plus feature oligonucleotide. So they took 70 base pairs of various sequences from every known mammalian virus, as well as microRNAs and other things. Each virus was represented by 10 or more features. So this is a little chip that has thousands of pieces of information, and it can tell us, and they looked at human influenza and avian flu viruses in a tiling type method such that they could identify viral subtypes, and I'll talk a little bit more about that later. But we can see both RNA and DNA viruses, and you can look at RNA detection if you put RNA on the array, and DNA detection if you want to put DNA on the array. We looked at RNA. It's highly sensitive for RNA, detecting down to 20 virus particles. So summary of what we found, I won't go into it because we did this array with 60 patients in Dan's cohort, and we did it twice. We did it at about 6 months apart, and we attempted to get the individuals at different disease states, when they're healthy, when they're sick, and we're in the process of analyzing. You can imagine the data sets our poor postdocs have to go through here to correlate the clinical data with the transcript. But what's important here is that we saw the top, they reported out the top 300 to 600 transcripts, and while herpes viruses predominated among the clonally rearranged in the lymphomas, the second most predominant virus was the Coxsackie virus family. So in the normal people, we found the endogenous retroviruses, which we all have in our genome, as well as ancient retroviral elements. Those are called lines and cymes, or jumping genes. They don't make full virus particles, and they're not known to be associated with any pathogenesis, though it's not unlikely that they can contribute to that. So we were excited about that, and so we decided, since we saw herpes virus, that what we'd do in the future was we would design, and we have done this. This one was available through the literature, but we and our colleagues, I say this we quite liberally, because this was all done at the National Cancer Institute in Frederick in their core facility for arrays, and they designed and built an EBV array, and they're designing for us and building at this moment an HHV6 array where you put every open reading frame of the virus on a chip, and you ask at any given time which genes. We heard a lot from Barton today about whether or not they were early genes or late genes. Which genes are expressed in the pathogenic states when the patient is sick? What does it mean, and which viruses are co-expressed and which genes lead to co-expression? Because EBV can talk to HHV6 and upregulate its expression. Retroviruses can do the same thing. There are retroviruses in a number of these patients. We've identified, and I can't talk about it today, but a novel herpes virus and a novel retrovirus in some of these patients, and we're feverishly characterizing those as we speak. So we've designed and validated the EBV array, and Jonathan's going to send us some samples real soon so that we can begin looking at these at this in his cohort, in his group 7. And then we're going to because of our database, we can do longitudinal studies and ask if clinical isolates differ among various patients, another way to subgroup what might be going on in the pathogenesis. Similarly, the immediate early region, which is the region that is distinct in the 6A and B, and likely involved in the differences in pathogenesis and cell tropism in these two, will be made as subarrays so that we can use those. We intend to use those. We'll license those from the government and use those in the diagnostic company, and we hope to put together some smaller chips so we can use this technology to ask which virus at any time. Now, that goes with the caveat that we know that the peripheral blood is not where these viruses are often expressed, but I won't talk about it today, but we have ideas there and data there to support how to deal with that. So let's talk about this clonal rearrangement. What this means, and I'll show you a picture of it later, is your T cell receptor genes are involved in recognizing all the antigens. And you will rearrange each receptor as you see an individual pathogen, which means when you look at these if you were to run them out on the gel, they would be different sequences, so they would run like a big smear. Now, the gamma delta chain rearranges before the alpha beta chain, and it's not the predominant T cell subset in your periphery. It's a rather primitive T cell subset, and one doesn't even know. There's actually no one knows really the mechanism by which the gamma delta T cell recognizes antigen, but it's one of the very innate. It's known to be upregulated in response to viruses. So Dan chose the patients over a course of the last 2 years. As with most of his patients in the cohort, they had an acute viral onset of the disease. They had lymphadenopathy and usually splenomegaly. He looked at 170 people. This is done by a multiplex PCR primer assay, which I will show you later, and it's available commercially from essentially any diagnostic company. It was validated worldwide. It's something you could do now if you suspect lymphoma, and that's usually the diagnosis that he uses to get that covered by insurance. Of that, if you looked at a normal population of 200 people, you might find one. Dan found 59 positive. They were all gamma. You can look for IgH, you can look for alpha beta. 59 of those were the T cell receptor gamma. 4 were the T cell receptor gamma plus the B cell receptor, and 1 was B cell receptor alone. So this is what the assay looks like. As I mentioned, you use multiplex primer pairs, which a company in the U.S. named InVivoScribe makes and sells. They mix these in 2 tubes, so it's pretty easy to mix them like cooking. And one primer pair set has these, and the other primer, or each of them have the black one. And these are mixed according to these 3 in one tube and these 3 in the other. They send with it controls. This is a T cell line, a B cell line, and the red line goes through the V gamma 11. So you see one single band. These are just molecular weight markers to focus your attention attention. And my postdoc likes to focus mine. But at any rate, you see that the Jurkat cell line, a well-known T lymphoma cell line, has the V gamma 11 rearrangement, as does the peer, but the peer also has the V gamma 9 T cell. Our patients, 1674, 1808, 1031, 1726, 1127. Interestingly, you see the smears here. That's not a positive. Right here's a positive. Right here's a positive. That's sorry, molecular weight. I'll focus here. You see when you see smears, that would be called oligoclonal. So you want it to be twice background over the smears. And we can read these with a software program which will give you a single piece. But essentially, of the 459 clones we looked at, they were all the V gamma 9, which is very interesting because that's a gut-associated gamma delta T cell. So they might be recognizing an enterovirus. But we haven't done any more of those work, and we're looking for a single base pair. If that was the exact same base pair of rearrangement, that would indeed be striking. So again, we wanted to look at, to subgroup these, we wanted to look at the rearrangements and the virus associations. So of the 59 patients who were positive, 13 were negative for HHV6, 41 were positive, and for 5, we didn't have the test in our database, and we didn't have time to do it yet. 19 of these were positive for 6, but not analyzed for the subtype. So we don't know if it's A or B there. Of the 19, 13 were positive for A only. There were some positive for A and B, and only 5 were positive for B only. So this lets you know that Dan is indeed right in that the major virus problem in his cohort is HHV6A. Interestingly, when we looked at all of the EBV and CMV in this cohort, essentially everybody had equal numbers of that, suggesting that it wasn't recognizing an EBV antigen or CMV. But again, these need to be done. Those are just suggestions. So here's what the data look like with regard to non-Hodgkin's lymphoma. And the reason I came to Nevada was because Dan reported at a meeting 2 years ago an extremely high incidence of a very rare lymphoma known as mantle cell lymphoma. He's got 6 of them in that cohort. There's 4 on this chart, but 2 have subsequently passed away, but they are, it's a rare. If you see 6 in a cohort of 50 people, you're talking about a cancer cluster when you're a virus hunter, and you're talking about looking for a new virus there. So we show you then the patients, all the patients in our database are patient numbers, and they're blinded to the investigators. Usually to me too, because the students don't tell me much. At any rate, we've typed them for HHV6 wherever we could, and you see the T cell receptor gamma clonality. That's usually wise, not to tell the PI. When we looked at the receptor clonal without lymphoma and with HHV6, you can see it here. It's a significant higher number. I mean, this is scary. Is this a precursor to lymphoma? Is this immune defect suggest you'll get lymphoma? So far, no, but we would like to think that this was something we could target and fix prior to the development of lymphoma, since non-Hodgkin's lymphoma is among the highest rising cancers essentially in the world, very rapidly rising. So these are the patients without HHV6, and the reason I say that is, do they have another virus? Is it enterovirus? Is it that novel herpes virus that we're looking for? We don't know. I will tell Martin that virus sits essentially right in the middle of 5 and 6, and we affectionately call it 5.5. So hopefully we'll have that fully analyzed. We were able to develop from one of these patients a cell line that was EBV negative, CMV negative, HHV6 negative, and lo and behold, it spit in our new virus. So if that's not luck. So at any rate, so then what we did then was we wanted to ask what was the cytokine and chemokine profiling in ME/CSF. And our long-term objective is to look at these 25 cytokines, chemokines in 150 of our ME/CFS cohort and in at least 250 controls, and establish the normal parameters. What's the range? These are normal proteins, and we need to look at the range so that we can make a proper assignment of subpopulations as well as accurate diagnoses if we're using patterns. So I go back to the innate immune response and just remind you that you can see patterns upon activation, and that an indication of what the pathogen is would be by its cytokine profile. But that's the innate immune response, and it's more complicated than that because as we've heard today, you need a functional adaptive immune response in order to have a fully functional immune system. And I pulled this slide from a Nature Medicine review looking at the development of a newly described subset known as the Th17 cell. Th17 cells make IL-17 as an effector cell. Interestingly, high levels of IL-17 are observed in the mouse, the multiple sclerosis model, that pathogenic EAE model. So suggesting Th17 is important in the development of neuroinflammatory disease. I show this here not to describe everything about it, only to note that we can look at the cytokines and chemokines that are involved in the development of these cells. IL-6 and TGF-beta are key regulators of this inflammatory cell population, as is IL-23, which is made by macrophages and antigen-presenting cells like B cells. IL-10, you heard about from Jonathan, is something that's been both up and down. And we see that in a lot of cases in cancer immunology, and we actually don't know if up or down is good or bad at this point in the development. So you can again characterize the various subsets of T cells by the cytokines they produce, and that the cells of the immune response all talk to each other. So you can quickly characterize functional cell subset deficiencies by their cytokine gene protein expression. So I'm not going to say a lot about this assay other than to say a suspension ELISA is where we take an antibody, to a very specific antibody to those 25 proteins, and we covalently link it to a 5.6 micron polystyrene bead that's injected with 2 different dyes, such that you can take a laser and read that bead and know which antibodies it's on, and then very sensitively and at a very big dynamic range, we can go from 1 picogram to 100 nanograms and detect protein differences. We can put all 25 of these in a single 50 microliter well of serum or plasma or cerebral spinal fluid or anything else, and there won't be any crosstalk, and they'll identify very specifically and very sensitively the quantity of each one of those. We did 91 of the patients and 121 controls. Statistics by the Mann-Whitney test of medians. We found 15 cytokines downregulated and 4 cytokines upregulated, and 11 that were not different between the groups. We sorted these out, and I'll show you what they look like in the next few slides. So what we see here, and our colleague Dennis Taub, who's at the Institute of Aging and has helped us interpret these complex data, has identified really that we see a shift of the Th1 and Th2 phenotypes towards a Th17 phenotype in ME/CFS. Interestingly, in our cohort, when we look at everybody versus normals, interestingly, you'll note here, well, it shows here that you have less IL-17, and but we have significantly and gross high levels of IL-6, that cytokine that drives the differentiation of this subset. I have other data to suggest why the protein level is low, and it's a little bit complex here, but clearly there is a Th17 shift in this population, and we need to understand how to modulate that back, and that's extremely important for tumor promotion. The T regulatory subset is downregulated. T regulatory cells are actually immunosuppressive. In cancer, they're a hindrance to immunotherapy because your body will tend to make a lot of these. This is the IL-2 receptor alpha. And your body will make a lot of these suppressor T cells to dampen the immune response. And then, of course, if you're doing immunotherapy in cancer, you're actually promoting the development of these, and that's a problem. So it's immunosuppressive. So what you want between autoimmunity and inflammation is a balance of these two, and you can see a shift. Again, IL-10 is upregulated by essentially 2 to 3-fold, and the Th1 cytokines, interferon gamma, IL-12, IL-2 as well, are also upregulated. So there was a feeling in the field that there's actually a Th1 shift in this disease when there's a viral insult. But we will show, and we'll hopefully publish a paper this year on this to show that that's actually a Th17 shift, and we have to sort this out. And I'll show you later on how we do sort this, because if you drop subgroups, you'll see this change to a very specific subgroup that is here, which is the cancer subgroup, and some of the other CFS subgroups that we haven't characterized according to defect in that group. So there are differences in the pro-inflammatory cytokines and chemokines. The interferons are essentially the same, which is quite surprising, because if we again sort this out in subgroups, we see patients, say 10% of our population, with extremely high levels of the type 1 alpha and beta interferon, where gamma interferon in our cohort is essentially never changed. GM-CSF, which is a very important stem cell granulocyte monocyte progenitor, and important in the inflammatory cascade, is upregulated in our population, as is the anti-inflammatory IL-1 receptor alpha is upregulated, and that's a natural response to seeing the pro-inflammatory cytokine IL-1 beta upregulated. Amgen is actually making a drug in rheumatoid arthritis to make more of this, because this actually will dampen the signal. It's a soluble receptor, and it'll dampen the IL-1 beta, and has been used effectively in arthritis. So all of these data suggest that, and we've talked a lot today about ME/CFS being an inflammatory disease, and I tabled these data from a review article that I could provide to anybody who wants it. And as you see the inflammatory disease, you see that you can actually profile, as I've suggested we can do in this disease, the chemokines, important chemokines. Chemokines are trafficking molecules. They tell cells where to go to the brain, to go to sites of inflammation. This is what they do. The acute respiratory distress syndrome, IL-8, this will bring in neutrophils, the activator of neutrophils, lots of oxygen radicals, peroxynitrites in neutrophils, a lot of production here. Bacterial meningitis, you can distinguish from viral meningitis based on the chemokine profile. Inflammatory bowel disease, psoriasis, atherosclerosis, we've talked a lot about today. IP10 is an interferon response protein 10 that responds to interferon, suggesting again a viral infection in this, that important in atherosclerosis. So what happened to the chemokines in our cohort? We see IL-8 just astounding, so 20-fold up in our patients. Important paper just came out in Blood last month showing IL-8 is the single marker for acute graft versus host disease. And if that didn't get the attention of our SAB chair, who didn't believe in any of this stuff when I came to Nevada a few years ago, he's now looking at our studies a bit more carefully. IP10 is upregulated in our patients, as well as the neuroinflammatory chemokines, MIP1 alpha, MIP1 beta. Rantes and neotaxins are very early responses, eosinophils and mast cells, and histamine and allergic reactions. When we sort these data according to just the patients with MCL, we identify a very different profile, again, suggesting that we can tell you what's going on based on the cytokine and chemokine profile. Here we saw that the differences between CFS, all CFS, there were no normal controls here. With and without mantle cell lymphoma were as shown here, where IL-7 was an important upregulated molecule in the disease, which is important in B cell development. Interestingly, it was just shown in Nature Genetics that the IL-7 receptor alpha was found by a genetic screen to be a predisposing factor if you have single nucleotide polymorphisms or abnormal IL-7 receptor alpha. They identified this in a huge screen where they looked at thousands of normals and used historical controls based on the large databases we're building, and we in this field should take advantage of using those, which we have in our group in conjunction with the NCI's Lab of Genomic Diversity. IL-7 is even more interesting because it had been described 2002 by a candidate gene approach in which investigators said that this gene was likely involved in multiple sclerosis, but nobody could repeat their work because they couldn't find it the next time. And the reason they couldn't find it the next time was the LOD score for this, the difference, it was 1.2. So you're looking at a difference over 1 of only 0.2. So if you don't subgroup and you're looking at everybody, the noise is going to mask the response. But this is extremely important because the mutation in this gene, in this cytokine receptor, makes the receptor soluble, such that all of the IL-7 is going to be soaked up in the periphery and is never going to signal this functional T cell, B cell to be developed. So other genes that were extremely important, we talked about IL-6. It's a key indicator that inflammation in that inflammatory T cell subset is likely responsible for promoting the tumorigenesis we see in our cohort, or at least partially responsible. These are the usual subsets, IP10, TNF-alpha, cell migration in the brain. So what happens when we look at individual patients at 2 different time points, as I tell you we did? This is a CFS patient with thymoma. We also had 5 thymomas in this subgroup. That's unusual. The thymus is where you educate T cells. There's something amiss, and if you've got a clonality, lymphocytosis of a gamma delta T cell, you might have other problems with regulatory T cell development. But when we looked at 2 different times in this particular patient, the blue indicates 2006, you see a dramatically different cytokine profile from 2008 with the development of the lymphoma. So again, we see this IL-8, and you don't see as much of the IP-10 later on in the development of the tumor, which suggests again that you can predict and you can use these profiles to look at dramatically different immune responses within the same patient and identify disease progression, or use this to monitor therapy to turn the cytokine back to the direction that you'd like to see it go. We have a lot more to do, but a patient with MCL, again, if you look at the cancer patients and you remember the last one, IL-8 and MIP-1 beta was a problem. No TNF, nothing else. Same thing with a patient with unknown lymphoma. It has not been characterized. MIP-1 alpha at both time points, IL-8. So what we draw this Venn diagram to show that in all CFS, there's overlap of cytokine and chemokine dysregulation, but that you can subgroup these patient groups according to clinical symptoms, which is indicated here by the different colors, and the cytokine chemokine expression profile that they have is distinct. So we talked a little bit about RNA cells, so I'll shift gears here. And RNA cell is a endonuclease antiviral pathway that John mentioned briefly, and it's been described for more than 15 or 20 years as being a major antiviral pathway which is dysregulated in this disease. And as I told you now, it's been identified in men that get inflammatory prostate cancer young. They have a defect in RNase L. They don't make RNase L, and they are susceptible to viral infection. A novel virus was identified in the prostate tissue using that viral microarray with in patients only with an RNA cell defect. We're all exposed to these viruses, they're susceptible. So we're looking, there hasn't been a lot done to identify the cellular phenotype. And what are the signals that upregulate and activate the only known substrate of RNA cell, which is 2,5A? What's happening here? We generally look at all the peripheral blood mononuclear cells and not an individual cell. We want to understand the viral evasion mechanisms of the viruses associated with CFS, and what that means is subgrouping. We're going to look at, are there particular viruses that are more prevalent when the RNA cell activity is low. Interestingly, in a large group, we have a constitutive high RNA cell activity, and I'll show you how that's determined in a minute. At the NCI, our colleagues Mary Carrington and Mike Dean are sequencing the RNA cell gene in our entire cohort to determine if the defects of the same sequencing protein gene defects are shown in both the cancer patients and our patients, suggesting why the protein isn't functioning. Again, we need to look at the cellular localization because we don't know if it's the neutrophil that's making the RNA cell the low molecular weight. It's a 37 kilodalton protein that Kenny Demerlier has described, and colleagues, Dr. Sadolnik at Temple University. This protein is constitutively active, and it essentially spins out and can't make any interferon. And what is the role of RNA cell in a chronically activated and inflammatory environment? So those are studies ongoing, and I'll just show you one example of what this looks like when we look at the whole cell, where we take a cell fresh immediately following isolation, we put it in Trizol, and we run it on a microarray, which shows you essentially the 28 and 18S ribosomal fragments. But if you look in lane 6 and lane 12, you see in this case where there's an excess of hyperactive RNA cell activity, you're getting degradation of your normal cellular RNA, and this isn't a good thing. So finally, I'd like to acknowledge the people that really have helped me build the program at the Institute, and really couldn't do anything without the work of the National Cancer Institute Cancer Inflammation Program. My mentor, Frank Rossetti, essentially does a lot of this work with his hands on weekends prior to when we got a lab up and running. So we send him samples, and I get to go to Frederick, Maryland every now and again, as I spent the last 2 days doing working in our lab that we worked in for 20-some years. Rachel Bagney was a student in the lab, just defended her PhD and graduated Sunday. And she made the viral microarrays. She's going to make for us the specific microarrays. Carrie Petro-Sadowski does all of the technician work. The National Institute of Aging is collaborating with us. Dennis Taub is recognized as the world's expert in chemokines. Couldn't do anything without our colleagues at the Institute for Viral Pathogenesis, Dr. Knox and Kerrigan. Actually also run a viral diagnostic company. You can get diagnostics from either our company or their company by PCR for all the herpes viruses we heard today, enteroviruses, a number of other viruses. They do retrovirus as well. At our institute, in our lab, essentially all of the work I showed you on the statistical analysis and the cytokine profiling was done by our talented and hardworking postdoc, Vinny Lombardi, and as Connie, who's from Wisconsin, would say, you got to love him with a name like that. And Darren White did this entire, he is going to medical school next year, but he did the entire database. I used to get a little frustrated because he'd sit with headphones all day long, and then I realized what he did when he went through those patient records in those samples in the freezer. And then to go into the executive team without whom the Institute wouldn't be possible. Nothing happens without Annette Whittemore. Like many of you here, she has a personal interest. Andrea, her daughter, became infected when she was 12. Dan was the only doctor, or at least the problem started when she was 12. The only doctor in Incline Village there has been treating her for over 18 years. The Whittemores had resources, recognized that many people couldn't get the kind of treatment their daughter did, and the two of them during the long treatment hours, basically in two 2005 said we've got to do something. Let's start an institute. As you've seen my plan, they found me only when I responded to Dan's plea for help at a meeting when he didn't understand the development of the mantle cell lymphoma, and I thought I would come for the summer and solve that and help him, and now I live in Reno, Nevada. So, but the plan is to not build another institute, but do something. Get people with expertise to do the tests they do and give them the tools to do it. Give them the right samples. Have a physician there who's looking for the right things. We want to train doctors, and we're serious about young people like Andrew coming so that they can, he's not going to last forever, and we need more doctors. We're struggling to hire doctors that are interested, and I know that's probably not surprising to you. Our scientific advisory board, I mentioned in passing, Bill Murphy, he's the chair of the Department of Microbiology and Immunology, and he was a colleague of mine at Frederick who happened to be in Nevada prior to my arrival, and he said to me, "What, are you kidding? You'll ruin your career." And fortunately, Frank Rossetti said, "I remember when there was no such thing as an NK cell," and Bill said, "Touche." So we're working quite well with Ken Hunter, who has a mouse model of autism and a marvelous immunologist there at the University of Nevada, Reno. We have on our scientific board the others that you see here. Nancy Klimas, a pioneer in the treatment at the University of Nevada. Again, Frank Rossetti with his knowledge in viruses and hematopoiesis and the cytokine modulators of hematopoiesis. I don't think we can go wrong. Suzanne Vernon has recently been promoted to the scientific director of the CFIDS Association. And Carl Ware, who we met at a meeting, is a marvelous immunologist. And I asked him at the end of the meeting, "What if I told you I had a human population for that mouse model you showed?" And he said, "I'd be interested." He emailed me essentially immediately, and he's been on the team ever since. And he has therapeutics developed and would love to run some clinical trials. So with that, I'll thank you for your attention.